So, can you hear me? In this talk, I am going to present work we have been doing in our labs on a family of new sequencing technologies called synthetic long reads, and on their applications to several problems in genomics, and in particular to genome phasing. This talk will mainly cover this paper that we published last year in Nature Biotechnology. This was a joint publication with Illumina, and in it we proposed one of the first commercial haplotyping technologies based on molecular long reads. And at the end of the talk, I would also like to discuss some other work that we have been doing with these reads in Metagenomics and in De-novo Assembly. But first, let's start with phasing. So phasing is the problem of determining the maternal and the paternal sequences of a given gene. It looks at a very fundamental aspect of the human genome, and it also is a very applied problem. It has both scientific and medical applications. As an example of a scientific application is studying the human epigenome. Here you see a region from chromosome 16 from a real genome in which there are transcription factor binding sites that are methylated in the mother but not in the father. And as a result, it could be that one copy will be expressed but not the other. Another medical example of phasing is HLA typing. You may know that immune response during organ transplantation depends on compatibility between a set of HLA genes. And these genes are often highly heterozygous. In this example, all the colored squares represent heterozygous SNPs. And so if we could know the two sequences, perhaps we could better match donors and patients for organ transplantation. The phase of a genome can be determined by looking at how variants co-occur together in sequencing reads. A sequencing read will determine the phase of the variants that it covers. And by connecting these variants at where they overlap, we can reconstruct haplotypes or haplotype blocks of a given length. Now currently, this approach does not work well on the Illumina platform because the reads there are simply too short to compute these to bridge the gap across neighboring heterozygous variants. Recently, however, this approach has started to be tractable thanks to new long-read sequencing technologies. And I personally became interested in this problem while working at a company called Moleculo that was developing one such technology. And what Moleculo proposed was a protocol for producing long, accurate, and affordable reads on standard Illumina sequencers via a pre-processing step. And later, several other companies launched similar offerings. Most recently, 10x Genomics is one such company. I will focus here on Moleculo, but the algorithms that I will propose hold in principle for any of these technologies. So how does Moleculo work? The high-level idea is to take DNA and cut it into fragments that are 10 kb long, and then dilute and partition those fragments across a large number of wells. Then the wells are given a unique barcode, and the wells are pulled together and sequenced using a standard Illumina machine. Then afterwards, we can use these barcodes to reconstruct from which fragment, to reconstruct the long fragments of origin using a computer. So in our paper, we proposed a phasing method based on this protocol. And our method involves several stages, but the first and the most fundamental stage is what we call local phasing. Local phasing is basically connecting long reads at their overlapping SNPs in the same way as I described on the earlier slide. And for this, we developed a new method that frames the problem as inference within a graphical model. And the method is more accurate than a previous state-of-the-art algorithm called RefHap. And because it's probabilistic, it produces useful confidence scores at each position. And I'm not going to go in more detail because this was the subject of an ECCB talk last year. So we ran this method on our molecular reads, and unfortunately we found that those reads by themselves were too short to produce haplotypes of a length that would be comparable to those of an alternative technology called LFR. And also, we found that almost 10% of variants were unphased because of various sequencing biases inherent to the technology. So when we saw these shortcomings, we went back to the drawing board and we came up with a slightly unusual idea. We decided to further improve the qualities of these haplotypes using statistical phasing. Now, statistical phasing is a common method for microarray data. And it uses the fact that we all share many common haplotypes, and these haplotypes can be inferred by looking at a large panel of sequenced individuals. In our paper, we propose a new phasing algorithm called Prism, which extends these earlier methods so that they can handle pre-phased blocks from local phasing. And we found that the Prism method worked really well, and there were two reasons for this. The first is that by taking pre-phased blocks, we can significantly improve statistical phasing accuracy because the pre-phased blocks eliminate many incorrect haplotypes that would otherwise be consistent with the data. And the second reason is that local phasing breaks down across long stretches of homozygous SNPs. But these are also regions that have high linkage disequilibrium, and therefore statistical phasing methods work better across these regions. And so the two approaches can naturally complement each other. And finally, this method is also probabilistic and again produces confidence scores that can be useful, for example, in medical diagnosis. More technically speaking, our method is based on the Lee and Stevens HMM, which is also used in most existing phasing and imputation methods like Impute2, ShapeIt, Mock, HappyUR, and others. And we augment this model with an additional set of variables that encode the phase of a block. And we determine the most likely haplotypes and the confidence scores using inference with belief propagation in this model, which is an algorithm that generalizes the Viterbi and forwards-backwards algorithms. So we tested this joint statistical and local phasing approach on this well-known trio that includes sample NA12878. And what we found is statistical phasing can increase the length of local blocks by almost tenfold, and can increase the phasing rate to as high as 99%. And at the same time, it can keep the error rate quite low, and the number of switches per megabase is less than one per is less than one in excuse me, in each of the three samples. We can also compare this to several similar technologies that were released at the time. These include two approaches that are essentially variants of LFR, which is Caper and Peters, and an approach that uses phosphate-based sequencing called by Kitzman et al. And notice here that the fragments that we're using based on Moleculo are shorter than those of the other technologies, sometimes as much as 10 times shorter. And we're also using less sequencing, sometimes as much as 5 times less sequencing. But nonetheless, we can do as well or even better on most metrics in this table. And this shows that this joint statistical and local phasing approach works well. And I think there's a higher level message that it's possible to take a new wet lab technology that has certain strengths and certain weaknesses and come up with methods that reinforce the strength and complement the weaknesses, and the result can improve the state of the art on an important problem. Now, we're also trying to do something similar on several other applications. One of these applications is metagenomics. We're currently trying to sequence the human gut microbiome using Moleculo. And we're also working on a tool called Nanoscope that will make this easier for users. There are two advantages of Moleculo for metagenomics. The first is assembly quality. On our data set, we were able to assemble more than 600 megabases of sequence into 50 kilobase long contigs, which is 7 times longer than what we had with Illumina. And several of these contigs were more than a megabase in length, including one that belonged to a species that was recently discovered using a method that involved pulling several hundred human samples. And this previous method identified the new species as a cluster of short contigs, whereas our method was able to assemble 2 megabase long contig from this new species. And the other interesting application of molecular reads is the following. We can map those reads either to the reference or to our assembled contigs, and we find there many variants that can be SNPs or short indels. And we have strong reasons to believe that these correspond to these corresponds to variants of bacterial strains. And what we think is even more interesting is that we can phase these SNPs in the same way as we phased the human genome in the first half of the talk. And we have an algorithm called Lens for doing this. And on our data set, Lens can find about 5,000 different haplotypes that span about 200,000 variants. And the N50 length of these haplotypes is around 22 kb, but some of them are as long as 120 kb. And this is an application that's possible with Moleculo and not with other methods that is much easier and with Moleculo compared to other methods. Finally, the last application we are thinking about is De-novo Assembly. Currently, in order to do De-novo Assembly, one would have to first assemble long fragments from short fragments and then assemble the genome from long fragments. But to do the first step, we need to cover each long fragment with something around 40x of short reads. This is inefficient, and we are thinking of how we can reduce this 40x internal coverage to something much smaller, hopefully something around 4x. And the intuition for why we think this is possible is the following. So consider a region of the genome in which there's a repeat sequence R that's flanked by unique sequences A, B, C, D. If we try to assemble this using a standard assembler, we would have this repeat structure. But let's say that we now have molecular data, and this sequence occurs as a fragment in one well that we're going to denote as red, and this one occurs in an orange well. Then we will have raw molecular short reads that will map to this region and to this region. And if we view it in the repeat structure, we will have red reads mapping to the blue unitig and orange reads mapping to the green unitig. And so we will be able to resolve this repeat structure. And we're currently working on an assembler that implements this idea. We have tried it on synthetic data, and preliminary results are looking promising. We seem to be doing reasonably well compared to the approach that starts with long reads directly. So this is the third and final application that I wanted to discuss, and I would like to conclude with two quick takeaways from this presentation. I think, so the first is that there are these new technologies that have been introduced recently, and they may have certain shortcomings, but overall they promise to make significant progress on some important problems in genomics. But in order to make this progress, we will have to come up with new algorithms for dealing with these technologies. And I think that this can be an interesting research direction, interesting both computationally and also one that will have important practical applications. Yes, this is the end, and I would like to thank all of these people that made this research possible. Thank Thank you very much.
Thank you. Any questions?
Great talk. Out of curiosity, how many barcodes do you need for the method to be efficient?
It uses 384 barcodes per library, and the results I showed here used 2 libraries, but in the paper we also have results for 1 library, and there the difference is the length of the haplotypes is about 100 kilobase shorter, and we phase 1% less less variance, and yeah, that's the difference.
It's kind of an obvious question, but how do you think the method compares to true long-read sequencing like PacBio or Oxford Nanopore?
Yeah, I think currently the error rate of PacBio is a little bit too high to do this. I think this is the main reason. In order to be able to call these SNPs, you would need reads that would be more accurate. But I have seen work that called the SNPs using another technology, for example, using Illumina, and then would phase them using PacBio. So I think that would be something that would work better than using straight PacBio. That's my
Yeah, I mean it's a bit of a price. I mean some people sequence the the whole genome. It just costs a lot of money to go into high depths to correct for the error.
Mm-hmm. Yeah.
So as a follow-up, have you assessed the accuracy of your assembly steps?
Yes, that's what we had on one of the middle slides, and we assessed this by looking at this trio. So we have NA12878 is the daughter, but we also have her parents, so we can determine the phase of about 80% of the her SNPs by using Mendelian inheritance rules.
I see. Any other questions?
Thanks. With respect to the metagenomics application, how does your approach influence the need for sequencing depth or coverage?
This influence. So you mean how much coverage do we need, or in this work we used we had 2 samples, in fact. One of them used 3 libraries and the other one used 7 libraries. So we used a lot of sequencing. I think it's possible to get good results with 2 or 3 libraries right now, which would so in terms of assembled long reads, that's about 900 megabases per library.